Hi, listeners. Welcome to episode 94 of Cell and Gene, the podcast. I'm Erin Harris. Christopher Horan from Artiva Biotherapeutics joined me for this one. Chris is Artiva's Chief Technical Operations Officer, and he and I get into the future of natural killer or NK cell therapies for autoimmune diseases. Artiva Biotherapeutics has been making waves in the biotech industry with their novel approach to cell therapy. Their lead product, AlloNK, is a non-genetically modified allogeneic NK cell therapy that's currently in clinical trials for treating certain autoimmune conditions, which we get into during our discussion. What sets Artiva apart is their manufacturing first approach, which allows them to produce off-the-shelf allogeneic NK cell therapies at a quite massive scale. It's an innovative process that has the potential to make cell therapies more accessible and manageable in community settings and addresses some of the key limitations of current cell therapies. Chris offers important information on the key factors that make NK cells promising for off-the-shelf cell therapy products. And he outlines the main hurdles in translating NK cell therapies from hematological cancers to autoimmune diseases, along with some other really important autoimmune disease-specific information. All right, so here's my conversation with Artiva Biotherapeutics' Chris Horan. Enjoy. All right, Chris, thanks for your time. Welcome to Cell and Gene, the podcast. Thanks for being here.
Thanks. It's great to be here.
So tell us a little bit about Artiva Biotherapeutics.
Yeah, so Artiva Biotherapeutics was formed about 5 years ago, and it was formed to develop technology from Green Cross in Korea. GC Cell has been working on cell therapy R&D for well over a decade. And so this was an opportunity for us to kind of leapfrog with that technology and bring it to the clinic. And our first clinical program was in non-Hodgkin's lymphoma. About 2 years ago, we pivoted to autoimmunity. And so now the company is laser focused on autoimmune indications. We had a successful IPO in July of 2024. And so we're well positioned to execute over the next couple of years.
Excellent. Okay. And that's actually what I want to continue our conversation on here now. Regarding autoimmune disease, so for example, I just finished up an article for Cell and Gene for an outlook for 2025 that focuses on all of the areas for non-oncology indications that will be a focus in 2025, and certainly autoimmune is one of them, which is perfectly timed for this conversation, which is great. So from your perspective, though, what are the main limitations of CAR T cell therapy, particularly for autoimmune disease?
Yeah, it's a great question. And it's, I think, a key differentiator for Artiva. CAR T therapy is fantastic, right? It's been very meaningful for oncology patients. But as an autologous therapy, it has limitations from a scale perspective. It's really good for targeting the thousands or tens of thousands of patients that are in need. But as has been seen over the last decade since the launch of the first CAR T, there is a capacity constraint and a cost factor that would, in my view, keep it from really being the answer for hundreds of thousands of autoimmune patients. So that's one aspect, is scalability and cost. Another factor is safety. In a third or fourth line oncology setting, the risk-benefit factor that a patient is willing to accept is very different than somebody in their 30s who might have lupus and is looking forward to the next 40 or 50 years. So the issues of CRS, of ICANS, of potential long-term malignancy concerns are present in CAR T. But as we'll talk about, I think in this discussion, we have some benefits there with our AlloNK product.
Yeah, and that's exactly what I want to talk a little bit about next, which is in fact NK cell therapy. Before we get specifically to Artiva, again, I'd love to get your kind of broad perspective on how does NK cell therapy address some of the challenges, faced by CAR T treatments? And then on top of that, what advantages do NK cells offer over T cells in terms of safety and efficacy for autoimmune disease?
Right. So NK cells are one of the things that attracted me to join Artiva 3 years ago. And I think it's because of the inherent kind of benefits and safety associated with NK cells versus CAR T or T cells in general. So we talked about one, I think the risk of safety issues, especially CRS, are present with CAR T in part because, there's a risk of expansion in the body. NK cells do not further expand in the body. They stick around for about a week and are able to effectively do their job. And in our case, the mechanism of action is ADCC. So that's antibody-dependent cellular cytotoxicity. We use a monoclonal antibody to target the NK cell, but there's just this inherent safety. And we think that that's really beneficial for NK cells in general, especially as we look at indications beyond oncology. The other thing that really attracted me to join Artiva is just allogeneic in general. I'm a big believer in off-the-shelf products in terms of their promise to serve a broader patient population at an affordable, cost to society. I come from the biologics industry where, that is the model, make to inventory and off the shelf. And so I have just, a kind of a personal attraction to allogeneic, and certainly NK cells are providing, potentially a path towards effective and safe allogeneic treatments.
Sure. And so beyond, say, scalability and, of course, affordability, what are the key factors that make NK cells promising for the off-the-shelf cell therapy products that we're referring to?
Yeah. So again, we're a manufacturing-first company, which comes out of the fact that we're leveraging over a decade of R&D from GC Cell, and one of the things that they did really well was figure out cryopreservation. So NK cells can be cryopreserved effectively. We cryopreserve in an infusion-ready media. And because of our long history, we already have 4 years of shelf life. So the stability of the product over a long period of time, the shelf life really enables off the shelf. In addition, there's no HLA matching requirements, right? So it is truly just an inventoried product that can be used by all patients. And the ease and convenience at the bedside means that there's no additional, kind of manufacturing manipulation at the clinic or the hospital before administration. So these are all hallmarks of off the shelf, and why, even though NKs are cell therapies, they have a lot in common with biologics from a supply chain and business perspective.
Okay. We talk a lot about, especially on cell and gene, NK cell therapies from a hematological cancer perspective. But, what would you say are the main hurdles in translating NK cell therapies for, hematological cancers to autoimmune disease?
Yeah, so this was a really light bulb moment for us in the sense that we had been conducting a trial in non-Hodgkin's lymphoma. We've disclosed some of the results of that phase 1 trial where we were seeing CAR T-like results, so really great efficacy and especially safety. And safety had so many benefits in terms of expanding the potential treatment centers to the community setting. So we saw this activity in NHL. We also have a partnership with AFAMED where they're exploring it in Hodgkin's lymphoma. And so we were starting out in hematological oncology, and we were seeing great results. When the Schett paper came out, we realized that allo-NK is really competitively differentiated for a deep B-cell depletion, which is what the immune reset trigger was all about. So our product, as I said, works on ADCC, and we have demonstrated in the NHL trial that the B-cell depletion in those patients is extremely deep. And it really is kind of supercharging the efficacy of the combination with the monoclonal antibody. So in the NHL trial, we combined with rituximab, and we saw differentiated results versus rituximab alone. And so that really triggered a pivot for the company about 2 years ago to have a laser focus on autoimmunity, because we believe so strongly in the hypothesis that was demonstrated in the NHL trial and can translate, we think, very effectively in a broad range of B-cell mediated autoimmune disease.
Good. Okay. A minute or two ago, you mentioned manufacturing first, and I want to go back to that. Yeah. Talk to us about Artiva's manufacturing first approach. What is it? How does it enable scalable NK cell production, and just sort of the mission behind manufacturing first?
Happy to. And again, this was one of the things that attracted me to join Artiva selfishly as a manufacturing and supply chain person. When I started interviewing and saw a front and center on the Artiva website that Artiva is a manufacturing first company, it really resonated with me, because I know from experience that a lot of cell therapy companies end up getting tripped up by CMC, by manufacturing. There are challenges that are just time consuming. I have no doubt the industry will solve those challenges, but for a startup that's on a timeline that has a cash runway constraint, sometimes that becomes really critical. And so manufacturing first starts with, again, the GC cell work. And some of this is iterative research that takes time, and we benefit from essentially a 10 to 15 year head start, even though we're only a 5-year-old company. I'll give a couple of examples of that. One is that after much experimentation, GC cell landed on cord blood as the ideal starting material for NK cell expansion. They looked at adult peripheral blood. They looked at iPSCs. Once they focused on cord blood, because it's naive, right? It has better donor to donor consistency, or it enables better donor to donor consistency. They then wanted to make sure they could expand it in a kind of commercial scale. So they engineered a proprietary feeder cell that is actually designed for cord blood NK cell expansion. And so we have a really robust process. I would say it's more like a registrational process from the moment the company was formed, because of the work of GC cell. And so that process, the quick math is that we take a cord blood that's pre-selected for high CD16 affinity and a KIR-B haplotype. As you know, the cord blood banks in the US alone have hundreds of thousands, if not millions, of cord bloods available. We ask the top cord banks to pre-select. So there's an ample supply from a starting material perspective, which is really important when you think about the future potential, scale of an autoimmune indication. So we pre-select the cord blood. We then grow that. We obviously select out the NK cells and grow that to create a master cell bank. It's really a kind of frozen intermediate cell bank. One umbilical cord can yield 50 to 80 master cell bank vials. We then freeze those. They're cryopreserved. They have long shelf life. When we want to make a drug product lot, we then thaw one of those master cell bank vials. And in a 50-liter bioreactor, we expand that again to create the drug product batch. Each drug product batch yields 80 to 100 drug product vials. So there's more than 4,000 vials for each umbilical cord blood unit. This is really, at launch scale already. So many cell therapy companies, really have a challenge where their initial phase 1 trial is done at a scale that can't even support a registrational trial. And then they have a lot of CMC work to do, a lot of comparability, from analytics and just an FDA kind of discussion perspective. We have the benefit, because we're manufacturing first, of having really a head start on those things. So we're able to produce, again, 4,000 plus vials per umbilical cord. to give you a sense, our current trials in autoimmunity, the dose is 3 vials. So, well over 1,200 patients for each umbilical cord that we produce drug product from. So it's really a massively scalable process already. There is opportunities to further scale it. If we are looking at success and commercial launch, we can go to a 200-liter bioreactor. The fact that we're in a bioreactor also speaks to the industrialization of the process. So it's almost entirely enclosed process today. And we're really pleased with where we're at from a maturity standpoint in the process. We have our own facility in Sorrento Valley in San Diego. We brag that it's a very modest facility. So from a capital perspective, this manufacturing first approach has enabled a process that's really very efficient from a capital perspective. The footprint is only 9,000 square feet. And in just a couple of sterile suites, we're able to yield that type of output. And we believe this is a differentiator and a competitive advantage.
Certainly, the efficiency is remarkable.
Can I mention one other thing, Erin, that I forgot to? Our lead candidate, AlloNK, is non-engineered, right? It's unmodified. So this is a big advantage as well. We also, in our pipeline, have CAR-NK assets. And we know from our own experience that the CAR transduction step with lentivirus is very painful from a yield perspective. So this product uniquely does not suffer the yield losses that any CAR-NK or CAR-T would suffer. So it's another reason why we have this massively scalable process.
Certainly, and I'm glad you brought up AlloNK, because that's one of the things I wanted to talk to you about, and perhaps this is a good time to do that. How does the therapy differ from other NK cell therapies in development?
Right. Even among NK cell therapies, I think we're differentiated. So certainly all of them are allogeneic, but there are very few, maybe a handful, that use cord blood as a starting material. Most, if not all, are CAR transduced. Many are using iPSC as their technology platform. So cord blood starting material, unmodified, no CAR transduction, are key differentiators for us for AlloNK versus other NK cell products that are competing in this space.
Remarkable. Okay. Can you talk to us about any early clinical data or ongoing trials, that are continuing to demonstrate Artiva's NK cell platform?
Briefly, I can just reiterate that we had this phase 1 NHL trial. The results we've communicated in the past, so about 45 patients, and CAR-T-like results in terms of both efficacy and durability. Which is fantastic, and I think kind of maybe surprised folks, because on the surface, AlloNK seems like a very kind of low-tech product, but yet this combination with an established monoclonal antibody with the NK cell boost has really demonstrated incredible results in the clinic for NHL in that phase 1 trial. So that's really the proof of concept. Again, I'll emphasize safety. We didn't see, the same level of risk in CRS. We had no ICANS. We had minimal hospitalizations, which really reinforced our view that this allo product can be safely administered in the community setting. And again, this was like the proof of concept for the pivot to autoimmunity, because these are really, in our view, very important in the autoimmunity setting. And I also mentioned Affimed. They have an ongoing program in Hodgkin's lymphoma in combination with their NK cell engager, AFM13. And they've seen very promising results as well. And I think announced recently that they have RMAT designation now from the FDA based on those results. So those are trials on, the Affimed trial is ongoing. The pivot for us has been to autoimmunity. We now have, enrollment execution as our priority, and we're expanding globally. So we've guided to preliminary data in the first half of 2025.
Nice. Okay. Just a couple more things I want to talk to you about before we wrap up. NK cell therapy evolving in the next, 5-10 years or so. I mean, even on Cell and Gene, we're doing a Cell and Gene Live later this year, dedicated solely to the evolvement of, the evolution of NK cell therapy. So, from your perspective, where do you see the field evolving to in the next few years?
I think there's a lot of promise in cell therapy in general. I think there's obviously promise in auto CAR T and the advancements happening in that space, also in allo CAR T. I still am a fan of NK cell, the future over the next 5 to 10 years, for some of the reasons we've talked about. The inherent safety is a big one. I think safety is top of mind for folks, especially in the autoimmune setting. So I think you'll see that Artiva and our peers will continue to make progress in both the CMC, scalability and process robustness, and also on the clinical development side in improving the efficacy and safety in the clinic. I really do think we're going to see approvals for NK cell therapies in the next 5 to 10 years. And, we're planning on success for allo NK, of course.
Certainly. And you did briefly touch on Artiva's roadmap for the first half of 2025, especially. But to wrap us up, tell us a little bit about what you're going to do what's Artiva's doing in 2025.
Absolutely. So we had a successful IPO in 2024. So that has funded us to really be laser focused on execution, as I said. So execution for Artiva, both in 25 and 26, is all about clinical trials. So we're expanding the sites. We're going global with our clinical program. Enrollment has been a competitive landscape for cell therapy companies, and the company is really focused on success in the clinic in terms of enrollment. That's one. I should mention one of the benefits for Artiva is this is what the company is working on, right? So we have almost 100 folks who are all pulling in the same direction for allo NK, and that prioritization is super powerful in terms of making sure we're all aligned and working towards a common goal. From a CMC and technical operations perspective, our priorities in 25 and 26 are several-fold. We've proven the San Diego facility. Last year, we made 18 GMP batches. This year, we're going to be upgrading the facility to make sure that we're ready for the next phases of clinical trials. We're also going to be implementing some process improvements. And over the next several years, the roadmap towards BLA means that we have, as all companies do, work to do to mature the process and the analyticals, the assays. And so there's a full plate of goals in 25 and 26 related to maturing all of those so that we're ready for the BLA filing and not on the critical path. That's always my goal in tech ops and CMC.
All right. Well, that brings us to the end of this episode of Cell and Gene, the podcast, featuring Artiva Biotherapeutics, Chris Horan. Chris, thank you so much for your time and your exceptional insight. It was great having you, and really appreciate everything you offered for our audience today.
Thanks so much, Erin. It was great to have the opportunity.
Absolutely. Listeners, be sure to visit cellandgene.com, and we'll see you next time.